Once again, thanks to lab, right? This is not an unfamiliar topic. Also, to some degree, thanks to mitosis, right? This is not an entirely unfamiliar topic. Since the stages or steps of meiosis are very similar to the stages and steps of mitosis. Okay, so one of the goals, as you'll find, and I mentioned this on our avatar video for this week, or one of the major pitfalls, almost if you will, is really making sure that we flesh out the differences between mitosis and meiosis, because there are so many similarities. And while that can help us, it can also run the risk of hindering us in a lot of ways by not being able to tell the difference or knowing where we are if we're looking at a stage. Okay, so like before, when we were going through mitosis, okay, we're going to start through pretty much the same stuff. We're going to go through important vocabulary, which again, largely is going to be very much similar or familiar to you. We want to focus on the function of meiosis. Sorry guys, I have a frog in my throat. Right, and then think about how does that function, okay, help guide us or remind us of what the outcome of meiosis should be. Okay, so we can use that as we go through our next step, the stages. Okay, and so remember, meiosis is a little different than mitosis, right? Because we have two. Meiosis 1 and meiosis 2. So we want to think about what's happening as we go through all of the stages. And why is that? Why is that happening? And then when we're done, we're going to talk about what are we actually doing with the outcomes, right, our daughter cells of each of these. Why is it so important? Okay. Let's go through some important vocabulary. Some of this you've been using all along and was largely introduced to in that flipped classroom scenario, those videos in lab. Okay, so gamete. What is a gamete? A sex cell. Good. Okay, so we have two kinds of sex cells. Okay, the egg, which comes from a female. This is the female gamete. And we want to remember when we're talking about gametes, these are haploid. Okay, so then if the egg comes from the female, the sperm must come from the male. Okay. So, male gamete. Also haploid. Okay, so when we use the term fertilization then, what is this talking about? And more specifically, when the gametes actually combine, right? So good. Right, so the event when the egg and sperm first combine. Right, 1 + 1 = 2. Right, so what's a zygote? A fertilized egg, right? Right, so this is the term we first use to refer to that individual, that first diploid individual after that fertilization event. Okay, so it's not a lot of cells, it's very small. but it is now no longer haploid, right? We're not talking about gametes anymore. Okay, so we're truly diploid. We did egg plus sperm equals zygote. And now I know we've heard a lot of these words before, and many of them are intuitive. We just want to make sure that we're very clear that we're using these terms correctly and appropriately. In a lot of cases, that we're assigning this haploid and diploid terminology correctly. So any questions about any of that? Shiny. Okay, so let's think about the function then of meiosis. All right, because this is a bigger piece here. This drives a lot of the drama, if you will, of what's happening, of what's going on in meiosis and why it's so different than mitosis. So what is the function of meiosis? What's the function of mitosis? Growth and repair, right? Right, so we thought about growth and repair. Okay, the key thing is if I'm going to repair a spot on my body, I wanted all my cells to be the same, right? I wanted nice, smooth arms. So what's the function of meiosis? Right, so I want genetically different organisms by what process? Reproduction. Okay. There's really important pieces here. Okay, so reproduction is the process by which this is happening. And one of our goals then is to get diversity or differences. That's a nice bubble. So we've talked a little bit about why this diversity is so important, right? So if we think about having a no diversity but still doing the reproductive pathway, right? So I'm going to clone myself, right, using, say, the same process as mitosis. Right, so I would just end up continuously reproducing exact copies of myself. I could have a whole city filled with Dr. Arnett's. Very excited. Okay. If we imagine the problem with that concept, okay, so if, for example, a disease, completely hypothetically, that targets the respiratory system were to come through, right, and I, perhaps completely hypothetically, am horrifically asthmatic, okay, what would be the consequences of having a city cloned exactly like me? So I'm getting killed off here, but certainly I would be not terribly tolerant or resilient to this disease, right? I would be more likely to get it, I would be more likely to get sick and killed off horrifically. Okay? As opposed to having a population that is very diverse, where you would have some individuals that are more resilient and more tolerant to this particular disease. So diversity has a lot of benefits, right? Some individuals are more resilient and more tolerant to some situations than others. And this goes for all things, right? Drought, coldness, right? Different types of diseases. Each individual genetic makeup makes you more or less resilient and tolerant to different instances. So having a good mix makes the population stronger as a whole. All right, so that's our goal. Different. We also have another problem, remember. If we think about mom, is mom diploid or haploid? Are you diploid or haploid right now? Diploid, right? What does diploid mean? I'm going to ahead of myself. Do we just have this on the next page? We do. We're diploid, right? All individuals are diploid. Right? So that means 2 copies. Where do your 2 copies come from? One from mom, one from dad, right? So here's mom. Woo! How did I do that? That's a new one, isn't it? Didn't swipe away, it just... So here's mom. She's diploid. She has her 2 copies. Okay. She's 2 copies, 2 sets. She got 1 set from her mom, okay, grandma in this instance, right? And 1 set from her dad. Okay. Same thing with dad, right? He's also diploid. We've got 2 copies. Okay, so all fully formed organisms are diploid. Plants, ignore plants. Plants are super weird. Right, because we all have a mom and we all have a dad. So we have 1 copy from mom and we have 1 copy from dad. Does this make sense? Yes? Okay. Same thing for a good baby, right? Baby's a fully formed organism. Okay, so what should baby be? Diploid, right? So baby should be diploid. Baby got 1 copy from mom and 1 copy from dad. This is where we get weird with meiosis, right? And we can't have 2 plus 2 equal 2, right? A number does not put the adding up. And we know that 2 plus 2 equals 4. Okay, so if we leave it as is, and I donate my whole set of chromosomes, put those in my eggs. Dad puts his whole set of chromosomes, put those in his sperm. And we just let that fertilize. We get mutant baby. Okay, all sorts of extra stuff. Weird arms sticking out of his head. Okay. Kid's gonna make fun of him at preschool. So we need to do something with our gametes to make sure that we can get 2 for baby. Or is back. So the other goal of meiosis is to make these haploid gametes, right? So my egg is haploid. Remember, haploid, literally think half. It has 1 copy of everything. And dad's sperm, running out of space, kind of put myself in the corner here, is also haploid. So it has one copy of everything. So that's what meiosis is doing, is it's going through and randomly selecting one copy of everything. And that's giving us some diversity because we're just going to randomly pick one copy of everything. And now we can mix one copy of this with a completely random subset from somebody else. 1 plus 1 does in fact equal 2. So we can have a normal baby, normal number of legs that's not going to get made fun of at preschool. So that's our other goal of meiosis, is we need to cut down the amount of stuff that we're putting in our cells so we don't end up with mutant babies. Okay, any questions about that? Are goals for meiosis quite clear? So we want to keep these two things in mind. Every step of the way, what can we do to increase diversity? And as we go, we need to try to decrease the amount of stuff that we have. Feel okay? Feel familiar? All right. So let's then go through, we're going to go through our stages, right? All 8 to 10 of them. And just like we did before, okay, and this is ideally a review because we did this in lab, so you have drawings of this. We did this with our kits. Okay. We still want to think what's happening in each stage. Okay. And now that we've... I'm ready for anything. Yes, I am. Miss my chenille sticks today. I knew there was an in-class activity in here. I get when we started up. All right, so we're going to go through this together. All right, so what I want you to do is we're going to start on our first page. I am definitely drawing, and you can see it. Still drawing. Make sure you leave yourself space. We're going to start with meiosis 1. And then, of course, we're going to do Prophase 1. So I want you, with whatever you're doing, to give yourself several colors. Okay, so let's start by drawing an interphase, so we know what our colors are going to be. Okay, first thing we need, of course, is a cell membrane. We're going to make that one color. Okay. You can make them the same as me. Might be easier. I don't care that you do, though. And I suggest you use like dots and dashes, or you can like scribble in dark for some of them. Just make it so it's obvious for you. Okay, for the next thing you're going to do, we can kind of make this way closer than we need because we're not going to put in any of the other stuff. What's the thing that holds all of our genetic information, our DNA? What protects our DNA? Nuclear envelope, right? Good. I'll lazy hand that because I did not give myself a ton of space. Okay, as we mentioned, our example cell is going to be an interphase. So am I going to draw chromosomes or chromatin? Chromatin, good. Now, I don't want this to get out of hand, so I still have to be diploid, correct? I need 2 copies of everything, one from mom and one from dad. Okay, but I don't want a million chromosomes, or we're going to be just drawing stuff all day, right? So, let's see. Where did purple? Okay. I have 2 chromosomes. Chromosome 1 has 2 copies, right? One from mom, unraveled. Different enough? Not in the slightest, Tara. And one from dad. Keep the key over here so we can check it later. Okay, then I'm just going to have chromosome 2. Same sort of dealio though. One from mom. And one from dad. All right, that's our starting cell. Everybody with me? So I don't care what colors you use or what textures you use, if you use pen and pencil, I don't give a hula hala. Okay, just make sure that all the different pieces are clear enough that when you go to do all the rest of the steps, it's going to be clear to you. Okay, so take a couple seconds, minutes, okay, to think about as we move into prophase 1. Okay, what needs to happen? Okay, so remember, for our cell, we would have had to have moved through S phase. Okay, so let's think through the things we need. Do I still have a cell membrane? Yeah, that's not going to go away, right? Let's go ahead and draw that. Okay, do I still have a nuclear envelope? Yep, I'm going to keep that for now. Give myself as much space as possible. Okay. What's happened to my DNA? They're nice and neat, okay? So meaning I've condensed them down, right? I don't have any of this fussy, stringy business. What else happened to them? Do I only have 1 copy? Right? No, we went through S phase, right? So I've doubled all of the DNA in there. I have 2 copies of mom, 2 copies of dad for everything. Let's see, where are all my colors here? Okay, well, I guess that's nice and neat for me. So that's why all of our chromosomes, remember, look like little X's, right? Because we've gone through S phase. We've got 2 copies. Neat. All right. What's the last thing that needs to happen? How are my chromosomes organized? What's different between meiosis 1 and mitosis here? And how does crossing over happen? Who crosses over? Which chromosomes? So who's mom's chromosome 1 going to cross over with? Dad's. Good. Perfect. This dad here. Right, so crossing over is going to happen, right? We need our pairs to hook up. I've made this cell ridiculously sized given how big I'm drawing all this stuff, but that's fine. So the first thing we need to do is find your exit buddy. This is getting progressively worse. All right, so remember these are called homologous pairs. Okay. Exit buddies. Okay, so one of the big differences, one of the most iconic things about meiosis 1 in general, but particularly prophase 1, right, is that the mom's version of any chromosome and the dad's version of any chromosome hold hands. Right? They pair up. We only see this in meiosis 1. So anytime you see this, you immediately know where you are. Now, the other thing we mentioned here is this is a great opportunity to get diversity in by doing crossing over. So remember, crossing over is when we're hooked up in pairs like this, okay, we're going to switch portions of our genes. Okay, so remember, genes are sitting on our chromosomes. So these guys have the same genes, right? They're just switching the versions that they have. Oh, you're going to erase the whole thing on me. Yep. So how about instead we I'm just going to color over it and hope it works. Which is exactly what I was doing, obviously. Kind of works. So I kind of always imagine this as like 2 little kindergartners running around on the playground and they switch mittens. All right, so I had pink mittens, Jasmine has blue mittens. Okay, we're running around holding hands, and I give her one of my pink mittens, and she gives me one of her blue mittens. So now I have a pink and a blue mitten. So this is all the stuff that's unique. What else has to happen in prophase 1? What else happened in prophase of mitosis? We put things in boxes. What else do we need? Yes. Our centrosome things. What color am I already not using extensively here? our movers. You don't forget, a lot of the stuff that we did in mitosis is still going to ring true. I put things in boxes. Okay, I still need to hire my movers. Okay, any questions about Prophase 1? Okay, let's take the rest of the time that we have to work through to the end of Meiosis 1. So from here, right, we want to figure out, I have a picture, this will be fun. Be rude. Are we recording, my dear? All right, so we left off, we had been working on meiosis. Okay, so we had our interphase, which I'm not going to redraw, you all have it in front of you. And we had done Prophase 1. Okay, so we're going to slowly work forward, continuing to add on. Okay, I'm not going to go through all of the stages, but I want to make sure we have some important steps along the way. I'm going to just hit some important stages to make sure what you guys have, okay, is what's going to get us into meiosis 2 as appropriately. Okay, so let's draw. Red, looks like red. Let's draw Metaphase 1. Metaphase is that we remember and we think about our checkpoints, right? As the cell control, we know Metaphase is a really important phase, right? Because if this goes wrong, everything from here on out is going to go wrong. Okay, so do I need my plasma membrane for Metaphase? Yes, no, yes. Okay, put that bad boy in there. Okay, what about a nuclear envelope? Should I draw that? No, right? So we lost our nuclear envelope. What phase did I lose my nuclear envelope in? Prometaphase. Right, so I still had it in prophase, and we lost it in prometaphase, right? Okay, so I have my homologous pairs. Right, we made homologous pairs in Prophase 1. That was like our big, special, unique shtick. Okay. Do we stay in homologous pairs or do we break them up? We stay in our homologous pairs, right? So if we go to all the trouble to make homologous pairs, we're going to stay in them. Okay, so the homologous pairs, let's make an imaginary line here, are what are going to line up against this imaginary line. Okay, now, the way they line up against this imaginary line is random, right? There's not a mom side and a dad side. This is the other way that we're getting diversity, right? So ideally you've drawn this already, right? Remember we're supposed to come completed. I'm going to randomly, maybe not so randomly here. Mom's red and green, so I'm going to alternate for the sake of trying to get some diversity. So here's mom's chromosome 1 and dad's chromosome 1. I forget we had the crossing over event. That's still there. And we don't lose pieces that we'd already done. All right, mom's chromosome 2 is with gray, it looks like. Dad's chromosome 2, right? And they also had crossed over. Maybe lazier, but the messiness is not intentional. All right, so we've lined up against the middle randomly with our homologous pairs. Still have our crossing over. We're still missing something. What are we missing? Our centrosomes, right? Our movers, and where are they? On the opposite sides of the cell. Right, and they should be attached. These are centrosomes. Attached by spindle fibers. Okay, so again, as we're thinking about metaphase 1, or anything in meiosis 1, the way we know we're in meiosis 1 is because we still have these homologous pairs, right? So anytime we see homologous pairs, we know we have to be in meiosis 1. All right. Let's pop over to telophase. Pretend that those are more or less equal. My capacity for drawing. There's my cell membrane. I've started the process of cytokinesis. Okay, so started to pinch those two cells apart. Do I need a nuclear envelope in telophase? Yes, right? So my nuclear envelope, now come back, right? I should have 2. One for each cell or daughter cell that I'm creating. Okay. These might look slightly different depending on how you sorted your metaphase. That's okay. Right, but so if I've sorted my metaphase, right, and then gone through anaphase, right, so we would just split. Okay, so my left-hand cell is going to get the left-hand set of chromosomes. My right-hand cell is going to get my right-hand set of chromosomes, right? We pull apart at where they were holding hands in meiosis 1. each still have a double copy of chromosome 1. And both still have some mom and some dad genes. And each have a double copy of chromosome 2. Each retaining parts of mom and dad genes. Okay, so because they're still double copies, because they both still have mom and dad genes, we're still diploid. Which is why we're stuck and we have to go through meiosis too. Remember, our goal is haploid. Now, on the plus side, do we have diversity? These two daughter cells look the same as each other? So do we have difference? Yeah, right? Do they look the same as the parent? No, right? So at least we're nailing that difference outcome, right? Okay, so let's go through, right, meiosis 2. Okay, so for right now, let's just start and let's go through prophase 2. I'm going to erase metaphase here. Bye, baby. As you go along, you should do it for both of your cells. My screen will not allow me to, so I'm just going to do it for one. So let's start meiosis 2. Go ahead and do prophase 2. Let's look at prophase 2 together, and then I'm going to have you finish up the road here. Okay, so let's start by drawing our cell membrane. Anytime we're doing anything cell, it's always the first thing we draw. Remember, we would want to draw both of these, but my screen is tiny. Okay, I'm going to draw my left hand cell. You can follow along with what I'm doing. All right, so I'm in prophase. I need my nuclear envelope? Yes. Remember, we keep our nuclear envelopes until prometaphase. And so, following up with what we sort of were talking about, right? If we had uncondensed our chromosomes, we would need to recondense these, right? Here are my chromosomes. Here's my chromosome 1. I still have that strip from dad on there, right? I maintain that piece of crossing over from meiosis 1. Okay, and here, the gray one. Here's my chromosome 2. It also maintains its strip from crossing over. This is a lot less to draw, right? We've got a whole lot less DNA now. And just like before, we still need our centrosomes, right? This feels pretty pretty familiar. It's pretty low key. Let's go ahead. Oh, I hope I just picture that. Righteous. All right. Let's finish out meiosis 2. All right, so we need prometaphase, prometaphase, 2, metaphase, 2, anaphase, 2, and telophase, 2. When I'm done with telophase 2, how many cells should I have? 4. Good. Okay, take a hot second to do this. Let's try doing metaphase together. All right, so I'm focusing on metaphase for a reason. It's one of the easiest stages to kind of get confused between all three. It's also a good one to really be able to tell the difference between all three. So here we are at metaphase. So we know how metaphase should be organized, right? Think metaphase middle. So if I'm going to move my chromosomes to the middle, do I want them next to each other in pairs? Or do I want them lined up tip to tip like kindergartners? Tip to tip, good. Now let's put our imaginary line in. It's always kind of nice to visualize. All right, so excellent. So we want these tip to tip. So the reason, remember, in meiosis 1, that they held hands and lined up as pairs is because they were the same chromosome, right? And as in they were both chromosome 1. They were both chromosome 2. Here in meiosis 2, oh, sorry, guy, you need your little booty. These are different chromosomes, right? If we go back and check our key, and they should be, this is a chromosome 1, and our gray one is a chromosome 2. Okay? So they can't pair up because they're not a set, right? They're not a pair. So they have to go tip to tip. So this ends up, it's his little booty too, looking an awful lot like mitosis, right? In fact, they've lined up exactly the same way as mitosis. So if I were to give you, say, a metaphase question, say what stage or what form of cell division were we in, mitosis, meiosis 1, or meiosis 2, you could pick out meiosis 1 right away because they were in homologous pairs, right? That's immediately how we can identify that one. So we can cross that one out. How would I know the difference between meiosis 2 and mitosis? How does this look different than our mitosis drawings? Okay, so one way I could tell is if the crossing over was obvious, right? Crossing over only happens in meiosis. How else might I be able to tell? Correct, right? So in meiosis, I've reduced the total number of chromosomes that I have here, right? Because my whole goal here was differences and haploidness. But if I were to do mitosis, right, all 4 of those guys should be lined up here because I want everybody to look exactly the same. So everybody has to have everything. Okay, so that would be how you would know we were in mitosis. Okay. I'm missing my centrosomes, my movers should also be attached by their spindle fibers. Let's put those in. Oops. Yeah, you get the idea. Ay, ay, ay. All right. There's a cell membrane, making two daughter cells. There's two poorly constructed nuclear envelopes. Remember, I've only followed the one cell line through here, but you should have followed both. So I've only got two cells here. You should have four, right? Because you followed both divisions from telophase 1. All right, so if we think about my metaphase from my last drawing, right, and yours may have variance depending on how you set your metaphase twos up. And my chromosome 1 should split. And that booty still is on her. My chromosome 2 will split. And one of those still has a booty. I had it on this side. Okay, so let's take a look at my final cell. I only have one copy now of each, right? I have one copy of chromosome 1 and one copy of chromosome 2. We are officially haploid cells. All right, so by the end of telophase 1, we'd already hit that diversity standpoint, right? Remember, we did a check. Those two cells did not look the same as each other, and they did not look the same as the parent cell. We'd accomplished our first outcome, which was differences. Now here at the end of telophase 2, okay, we've reached our second outcome. Now we still have that first outcome, right? These don't look the same as each other. None of the 4 of your cells should look the same as each other. We still have those differences in there. And now we have our second outcome, which is that we finally made haploid cells. Okay, any questions about the process that we did for our in-class activity or the 4 cells that you've created on your sheets in front of you or in your computer, depending on how you've done this? We can almost think of each meiosis stage as accomplishing each of our different goals, right? Meiosis 1, right, is set up to accomplish that diversity or that difference goal. Meiosis 2 is set up to help us accomplish our haploid goal, right, or dividing everything in halves. If we keep that in mind, we had 2 goals for this one, right, whereas in mitosis we only had 1 goal, so we've got 1 stage. Meiosis, we have 2 goals, so we have 2 stages. Okay. There is as always, and we finished that, right? And ideally yours should be completely done. I think I'm going to do this now. Jumping ahead. No, I'm not jumping ahead. There's a in-class activity Dropbox. Okay, yours should be beautiful and finished, so not in class, right? But as soon as you are done and you have time today, please make sure you upload that. Remember, if you're taking pictures, you have to paste the pictures into a Word document. And get that uploaded. It'll be unlocked until tomorrow at midnight, just like always. The thing. Okay. All right, so let's practice picking out the difference between mitosis, meiosis 1, and meiosis 2. Okay, so let's go to Menti. Many of you are already up, right? Remember, you can do this on your phone or on your computers. So our code is 43 38 35 2. Okay, so we're thinking of a cell that is diploid. That's number 2N is diploid. It should have 6 chromosomes. So I'm giving you what it looks like. Tell me what phase this is. Okay, let's look at the pieces that we have. Okay, first off, and we know how to identify meiosis 1, so let's just see if it's that, right? How do I know if it's meiosis 1? What was our big feature of meiosis 1? Homologous pairing, right? Good. Do I have homologous pairs up here? No, right? Oops. All right, so we know it's not meiosis 1. And that's one thing we want to remember. If I can't see homologous pairs, already narrowed down, 1/3 of our options. Okay, so now I know it's either mitosis or meiosis 2. I know this is metaphase, right? Because everybody's lined up in the middle. All right, so now I need to use the information that's in the actual question. All right, so what I need is this. Diploid equals 6. So that's telling me that I should have 6 chromosomes. How many chromosomes do I see? 3, right? 1, 2, 3. They're duplicated. It's gone through S phase, but it's 3 chromosomes. Everybody see that? Okay, so if I only have 3 chromosomes, okay, what phase am I in? Have to be in meiosis 2. Remember, if we're doing mitosis, the goal of mitosis is sameness, right? Which means you have to give half of everything to everybody. Mitosis has to have everything, which in this case would be all 6. Okay, since there's not 6 chromosomes there, no, it's not that one. Okay, so there's only 3. 3 is half, okay, which means I'm making that half a haploid. And my telophase here will make a haploid cell. That's meiosis 2. Does everybody see how I worked through that? Okay, any questions about this question or anything that we did today? Okay, when you're complete, back 2 rows, you can go. else, you can leave. Please have a good day. Looks like it's going to stay sunny. Everything okay, Jasmine?
Oh, yeah.